So I told you that we are discussing the how rate of photosynthesis can be calculated using this setup of experiment. This experimental setup is similar to respirometer, in which we calculated the rate of respiration. Using this experimental setup, we can calculate the rate of photosynthesis. How is the rate of photosynthesis calculated? You can see there is a beaker in which water is kept and light is source is kept beyond it. On the other side, you are keeping a test tube with a plant, aquatic plant, for example, Alodia, into it, and then it is filled with water, and a tube is connected to it, which joins this to a gas syringe. A scale is also placed, although we won't be making use of this scale. If we make use of this scale, it can be done as well. This will be similar to how rate of respiration is calculated, that you would note down how much gas is collected in a known time or how much the bubble moves. But in this case, more appropriate measurement can be taken using the syringe here. So what do? We have a stopwatch as well. We know that our entire setup will allow photosynthesis to occur. So once the setup is arranged, our gas syringe will be pushed to the lowest, which means that it will not have any gas collected at that time. What we'll do, we'll allow this setup to stay like this for 2 minutes so that the plant gets adjusted or acclimatized to the new environment. Now we turn on our stop clock, and this time is 0 seconds, and we allow photosynthesis to occur. Now what we can do, we can either calculate the time taken for a known volume of oxygen to be collected, that can give us rate of photosynthesis. Time taken for a known volume of oxygen to be collected, or else what you can do, known time may how much oxygen is collected, you can do this either. How much oxygen is collected in known time, or time taken for a known oxygen to be collected. So either way, rate of photosynthesis can be calculated. So one thing that you need to keep in mind is that this is an aquatic plant and you are keeping it in a non-natural environment. It's not its actual habitat. So you need to provide this water with a carbon dioxide source. So our carbon dioxide source will be hydrogen carbonate. Hydrogen carbonate will be hydrogen carbonate is basically a salt, and it is a source of carbon dioxide. So when we dissolve hydrogen carbonate or bicarbonate ions in this solution, what do we get? We get carbon dioxide in the water which is surrounding this elodia. So if we go for our standardized variables, what will be our standardized variables? Light intensity can be kept constant, keeping in mind that, for example, we are calculating the rate of photosynthesis at different light intensities. So what we can do, we can keep the light intensity constant initially. For example, we can use a 10-watt bulb, keeping it at a constant distance from the plant. And when we are going to the plant, we basically note down the mass of the plant so that its mass on electronic balance can be standardized as well. You will need it when you are repeating it. Now when you add sodium bicarbonate into it, which is hydrogen carbonate ions, when you add sodium bicarbonate, you have to weigh sodium bicarbonate so that only that amount is always used in this particular experiment. The amount of water needs to be kept constant as well. And what other else you are keeping constant? Either you are keeping the time constant and noting down how much oxygen gets collected in the syringe, or you are keeping the volume of oxygen collected in the syringe constant and noting down how much time it took to get that kind of oxygen collected in that gas syringe. Now you carried on this experiment on 10-watt bulb. What you can do, you can change the light intensity. Once this is done, you go to a 20-watt bulb. You replace it with a 20-watt light, then a 30-watt light, then a 40-watt light, then a 50-watt, then a 60-watt light. You are changing the wattages of the bulb. You are changing the power of the bulbs. So using the different power sources, you can calculate how rate of photosynthesis varies with different light intensities. You can repeat this experiment. You will state this, that experiment can be repeated using the same mass of all these things, the same mass of the same mass of sodium bicarbonate, the same volume of water, the same mass of Elodia plant, and keeping all the conditions externally constant. Why are we using the water beaker here? We are using the beaker of water to keep the thermal component of light from harming the plant or raising the temperature of the plant. Otherwise, the temperature would rise as well when we increase the light intensity, and that may damage the plant. So this is one thing that can be done. The other thing that can be done from the same setup is how much the rate of photosynthesis depend on carbon dioxide concentration. You can keep the light source constant and you can change the carbon dioxide concentration. How can you do that? For example, you set your experiment at 50-watt bulb or a 40-watt bulb. You will state that keep the 40-watt bulb at a constant distance from this setup, and then add sodium bicarbonate, for example 1 gram of sodium bicarbonate, and dissolve it in water and place the Elodia plant, leave it for 2 minutes, and then start the timer and note down the time taken for oxygen to be collected, known volume of oxygen to be collected. Then you can just repeat it. You can change the carbon dioxide concentration by this time adding 2 grams, then adding 3 grams, then adding 4 grams. You can change the variable of carbon dioxide by increasing the amount of how much sodium bicarbonate is added to water. But you have to make sure that all the carbon dioxide initially got used up. How is that possible? You use different setups. For every carbon dioxide concentration, you use a different setup, or otherwise the other thing could be you use a carbon dioxide probe. What is a carbon dioxide probe? It is basically an electrical device, electrical appliance. You dip it into the water and it gives you a digital reading of how much carbon dioxide is there in water. You can increase the carbon dioxide concentration accordingly. Then you can state that you can just go for and then draw a graph of these, or either you calculate the rate of using 1 upon time. We have discussed how rate can be calculated in respirometer as well. So this is the basic description of how this setup can be used to calculate the rate of photosynthesis.